Hi guys, NTA level 4. This is Human Anatomy and Physiology, session number 4 by Mr. Erick Daniel Simba. Kama tulivyo discuss kabla, we have already discussed about the introduction to human anatomy and physiology and the structural organization of the living organism, and also we have discussed about the structural types and the functioning of human cells. Now let us see the common disorders of human cells, and this below learning tasks. By the end of this session, every student should be able to define terminologies associated with cellular changes, to define terms cancer, tumor, and neoplasm, and to list the common types of cancer, and to explain characteristics of benign and malignant tumors, and to explain etiology and associated factors for tumors or cancer. So by the end of this session, every student should be able either to define, list, or explain these learning tasks as discussed below. By starting, let us discuss about definition of terminologies associated with the common cellular changes. Anaplasia refers to the loss of the tissue differentiation and the function that is a characteristic of most malignancy. Anaplasia ni neno ambalo linatumika kuelezea uwezo wa tissue kushindwa kudifferentiate and to perform specific function. Anaplasia is a condition of cells with poor cellular differentiation. This is commonly due to losing the morphological characteristic of the mature cells. Before, in the last sessions, tuliongea juu ya uhusiano kati ya juu ya anatomy and the physiology of living organism. Even in the cellular level, cell pamoja na cell structure yake pamoja na namna inavyofanya kazi, they are related, they are always related. Namna ambavyo cell inaonekana kiumbile pia na structure ile structure yake kiumbile morphologically na namna inavyo perform function, they are always related. So once cell lose the morphological characteristic of mature cells, pia hata uwezo wake wa kuperform specific function may be lost, and the tissue differentiation is from cellular differentiation. The cellular differentiation is the process whereby the cell become more specialized to perform specific function. So when the cell or tissue lost the ability to differentiate so that it can perform the specific function, is what we call it anaplasia. And this is a common characteristic of most malignancy. The word malignancy and the word cancer is synonymous. This is most characteristic of most cancer cells. When you say cancer cells and the malignancies, they are almost the same. So let us see another word, another terminology known as atrophy. Atrophy refers to the decrease in the size of the cells. When the matured cells, the adult or differentiated cells decrease in size, is what we call it atrophy. Atrophy refers to the decrease of the size of the cells with a subsequent decrease in the size of the affected tissue or organ. When the cells of certain organ or tissue decrease in size, and also the tissue or organ of that formed from those cells will also decrease in size. So that is why we say atrophy refers to the decrease in the size of the cells with subsequent decrease in the size of the affected tissue or organ. This is also refers to wasting away. The common form of atrophy or organ wasting away is muscles atrophy. Muscles atrophy is when muscles wasting away, and it's usually caused by a lack of physical activities. Kwa mfano ukipata labda shida and labda ukafungwa POP for the long time, ukashindwa kumove your leg or arm for the long time, unaweza uka experience mkono wako kuwa mwembamba zaidi au mguu wako kuwa mwembamba zaidi kwa sababu ya zile muscles zinakuwa zime waste away. So when a disease or injury makes you difficult or impossible for you to move an arm or leg, or so the lack of mobility can result in muscles wasting. So muscles wasting is from atrophy. So atrophy may occur due to immobilization of the organ. This decrease in the size of the cells with subsequent decrease in the size of the affected tissue or organ may be caused by either immobilization of the organ for the long time. So in summary, atrophy refers to the decrease in the size of the cells with a subsequent decrease in the size of the affected tissue or organ, and this is also refers to wasting away. And the common form of the atrophy is muscles atrophy, when muscles wasting away due to immobilization. But in organ level muscles zinapungua in size, lakini ni kwa sababu zile cells ambazo zinatengeneza muscles zime decrease in size. Yeah, let us see about the word or terminology dysplasia. Dysplasia, this is a bloody term. This bloody term refers to abnormal development of the cells within the tissue or organ, and may progress to neoplasia, tumor formation, usually malignant, or revert to normal if the causing factor is removed. So dysplasia, this is just a broad term compared to other terms ambazo tumeshazi discussed before. This is a broad term that refers to abnormal development of cells within the tissue or organ, and it can lead to wide range of conditions that involve enlarged tissue or precancerous cells. And the development of dysplasia is commonly in children and even young adults, and it can affect many parts of the body including skeleton. So this is just a broad term that used to explain the abnormal development of cells within the tissue or organs, and it may progress to neoplasia. Neoplasia, the word which is synonymous to tumor formation, and this tumor formation may be cancerous or not, and also may be reversed to normal if the causing factor is removed. Kama kulitokea na sababu ambayo ilipelekea dysplasia kutokea, if that causing factor ikiwa removed, the cell inaweza ikarudi kuendelea kuwa normal na zikaendelea kuzalishwa seli ambazo ni normal. So this dysplasia used to explain or to describe the abnormal development of cells within the tissue or organs. Hyperplasia refers to an increase in the number of cells of the tissue due to an increase in the frequency of the cell division. So wakati seli zinapokuwa nyingi kuliko kawaida, kwa sababu ya ile rate ya kujigawa, cell division imekuwa kubwa zaidi, the condition is known as hyperplasia. And the common form of hyperplasia is endometrial hyperplasia. Endometrial hyperplasia ni mfano wa common form wa hyperplasia, is caused by too much estrogen or not enough progesterone. These hormones, estrogen and the progesterone, they play roles in the menstrual cycle. Estrogen makes the cells glow, while the progesterone signals the shedding off of the cells during the menstrual cycle. So hormonal imbalances can produce too many cells or even abnormal. Estrogen is responsible kwa ajili ya kubuild up zile cells endometrial tissues ndani ya female reproductive system, lakini progesterone ni responsible kwa ajili ya shedding off ya zile kuta za female reproductive system. So imbalances of this hormone, let's say the high level of estrogen and the low level of progesterone, may result to production of too many cells or even abnormal cells. Here let us see the word hypertrophy. The word hypertrophy ni kijumbe cha neno atrophy. Hypertrophy is the increase in the size of the cells without cell division. Seli zinapokuwa zinajiongezeka kisize au kiumbile, lakini sio kwamba zinaongezeka in number, is what we call it hypertrophy. So and also the most common hypertrophy is muscle hypertrophy. Is a term muscle hypertrophy is a term for the growth and the increase of the size of the muscle cells. Kama tulivyoangalia kwenye atrophy tukatolea mfano wa muscles zinakuwa zinapungua, muscle cells zina reduce in size, maybe due to immobilization for the long time. Here muscle hypertrophy is a term used for the growth and the increase of the size of the muscle cells without the cell division. The most common type of muscular hypertrophy occurs as the result of the physical exercise such as weightlifting. Wale ambao wamewahi kunyanyua vyuma kama mazoezi, the more you are doing exercise, na the more the muscles zina build up, zinakuwa enlarged in size. So hypertrophy is when muscle cells increase in size without the cell division. And even this term hypertrophy is often associated with weight training. Pale ambapo mtu anafanya mazoezi kwa ajili ya ku grow up the muscles, what happens there, muscles zina increase in size. So the process, or when the muscle cells increase in the size of the muscle cell increase in size without cell division, the condition we call it hypertrophy. And also we have another term known as metaplasia. Metaplasia is a transformation of one type of cell to another type of cell. Metaplasia is a transformation of one type of differentiated cell to another type of differentiated cell. So wakati seli ambayo imekuwa matured na imekuwa differentiated ikachange kutoka kuwa aina fulani ya seli na kuwa aina nyingine ya seli, is what we call it metaplasia. So metaplasia is a transformation of one differentiated cell type to another differentiated cell type. The change from one type of cell to another may be part of the normal maturation process or caused by some sort of abnormal stimulus. Hii inaweza kawa ni normal physiology au normal maturation process of the cell, kwamba cell wakati imekuwa matured ni aina fulani ya seli, lakini during process of maturation ina change kuwa aina fulani ya seli. So that is normal, and we can call it metaplasia, but sometimes it can be abnormal and is stimulated by abnormal stimulus. The one kind of cell change to another kind of cell as abnormal condition stimulated by abnormal stimulus. Here let us see the definition and the common types of cancers. Tushamaliza terminologies associated with the cellular changes. So now let us see definition and the common types of cancer. Cancer is a group of diseases characterized by uncontrolled or abnormal cell proliferation. Cancer ni ugonjwa unakuwa characterized na uncontrolled or abnormal cell proliferation, kwa maana ya kwamba those cells ambazo zinakuwa produced au zinakuwa proliferated zinakuwa divided without control and they are abnormal. That is why tulianza kudiscuss juu kabla the cellular the terms used to describe the common cellular changes. Zipo changes ambazo zinaweza zikatokea kwenye maendeleo ya ukuaji wa seli ambayo pia inaweza kapelekea cancer. So cancer ni ugonjwa ni magonjwa ambayo yanakuwa characterized by uncontrolled or abnormal cell proliferation. Proliferation is synonymous to say that the cell division. The cell division and the proliferation are synonymous. So when cell division is not under normal control and those cells produced they are abnormal, the condition is known as cancer. When cells in a part of the body divide without control, cells in a part of the body divide without control, the excess tissue that develop is called a tumor or neoplasm. So neoplasm or a tumor occurs when cells in a part of the body divide without control. Seli zikijigawanya bila control, the excess tissue that develop due to that process is known as tumor or neoplasm. The word tumor and the neoplasm, they are synonymous. Ni maneno ambayo yanafanana. So the study of tumors is oncology. The study of tumors is oncology, and the tumors may be cancerous and often fatal or may be harmless. Tumor inaweza ikawa ni kansa na hadi ikamuua mtu kiasi ambacho ni ikaweza kumuua mtu, namna kansa ambayo is very dangerous. Lakini pia tumor inaweza ikawa harmless kama tu ni tumor ya kawaida ambayo si cancerous na ikapona kirahisi kuliko zile ambazo ni cancer. So when something to note, very important, when cells in a part of the body divide without control, the excess tissue that develop is called tumor or neoplasm. So ikitokea sehemu ya mwili seli zinajizalisha bila control, ile ile tissue ambayo inakuwa developed in excess as the result of the cells divide without control is what we call it tumor or neoplasm. And the study of the tumor is what we call oncologies. And the tumors may be cancerous and often fatal. Call this neoplasm or tumor may be cancerous and often fatal. Fatal ni kitu ambacho kinaweza kuua. So lakini pia inaweza zikawa harmless. So tumor au uvimbe unaweza ukawa ni wa kansa au unaweza usiwe wa kansa. Iko ni kitu cha msingi cha kuzingatia in this slide. Cancer is an unregulated proliferation of cells due to loss in normal control, resulting in unregulated growth, uncontrolled growth, lack of differentiation, local tissue invasion, and often metastasis. Cancer cells zinakuwa under unregulated cell division. Cell division inakuwa controlled by the automatic mechanism za mwili. Mwili una uwezo wa kawaida wa kucontrol the cell division. The cell division should not be in excess. Kuna kipindi cell division should be high and kuna sometimes cell division should be slow. So mwili una uwezo wa kucontrol the cell division. So inapotokea mwili ukashindwa kucontrol kwamba cells zikawa na divide without control and those cells produce the abnormal, the condition is cancer. So kwa maelezo, cancer is an unregulated proliferation of cells due to loss of normal control, resulting in unregulated growth, unregulated cell division, lack of differentiation. The cells produced zinakuwa dedifferentiated, and the local tissue invasion and the metastasis. Unregulated growth ni maana yake ile cell division inakuwa haiko under normal control. Zile cells zinakuwa zinajizalishwa kwa wingi kuliko vile ambavyo zilikuwa zinahitajika. Lakini lack of differentiation, zile cells ambazo zinakuwa produced zinakuwa hazina uwezo wa kuwa specified, kuwa specialized for the specific function. Before tuliangalia cellular differentiation is the ability of the cells to become specialized for the specific function. So cells ambazo zinapatikana kwenye figo ni tofauti na cells ambazo zinapatikana kwenye ini. Utofauti ule ni kwa sababu ya cellular differentiation. Cells za kwenye figo hazitakiwi kuwa kwenye ini na cells za kwenye ini hazitakiwi kuwa kwenye figo, and the differentiation of those two kinds of cells ni tofauti. In human body kuna seli zaidi ya aina 250. Seli zote ziko tofauti kwa sababu ya the process known as cellular differentiation. So seli suni imekuwa divided. Those daughter cells need to be matured and differentiated to perform the specific function. But in cancer cells, those cells produced zinakuwa zimelaki ile differentiation, so haziwezi kuperform specific function accordingly. And the local tissue invasiveness. Invasiveness is one of the most important characteristic of the cancer cells. Invasiveness is the ability of the cells or tissues ambayo imekuwa cancerous extend into neighboring cells na induce ile hali ya kuwa cancer. So neighboring tissues around the cancer cells zinapokuwa na uwezo, inapotokea zikawa induced ile cancer ikahamia mpaka kwenye tissue ambazo zinaizunguka ile organi ambayo imekuwa cancer, is what we call it invasiveness. But metastasis is also another important characteristic of the cancer cells. Metastasis is a spread of cancer cells to new area of the body, often by the way of the lymphatic system and the bloodstream. Once cancer cells may develop, zinaweza kuwa transferred from the place where the cancer started to another area in the body. Zile cells zinaweza zikabebwa kupitia kwenye bloodstream or lymphatic system, then zikafika sehemu nyingine zikaendelea kukua na kufanya damaging kubwa zaidi sehemu mbalimbali za mwili, kwamba cancer cell may undergo invasiveness and also may undergo metastasis. The term cancer, malignant neoplasm and malignant tumor are synonymous. They are similar. Ukisema cancer, ukisema malignant neoplasm, ukisema malignant tumor, or ukisema malignance, these terms are synonymous. They are distinguished from benign tumors by the properties of the differentiated invasiveness and the ability to metastasize. Tumeangalia hizi sifa 4. Cancer cells lazima iwe na hizi sifa 4 zote. Cancer cells zote zipo under unregulated growth control. Yani ile cell division yake haipo under control, under normal control ya cell division. Zenyewe zinakuwa zinajizalisha kwa wingi kuliko kawaida, lakini pia zimelaki differentiation. They are differentiated. Haziwezi kuperform specific function kama normal cells, lakini pia zina ile process wanasema invasiveness. Zinaweza kuextend to another neighboring tissues na zika induce ile hali ya ukansa kwenye tissue zingine zinazoizunguka. Pia metastasis. Once cancer cells zinapoingia kwenye blood stream au lymphatic system, zinaweza kuzunguka na kwenda kukaa sehemu nyingine ya mwili na kusababisha madhara kama pale ambapo ilianza kukua. So most benign tumors may be removed surgically if they interfere with normal body function. Benign tumors, they are not malignant tumors. Sawa? Malignant tumors, they are not benign tumors. These benign tumors, they are not malignancies. These benign tumors, they are not cancerous, lakini malignant tumors zenyewe ni cancerous. Ufanano kati ya malignant tumor na benign tumors ni kwamba zote zinatokea na uncontrolled cell division. Zote ni kwamba seli zinajigawanya bila control. Lakini malignant tumor yenyewe kinachoongezeka ni kwamba zile cells zimelaki differentiation, pia they can undergo the local tissue invasiveness, lakini pia they can undergo metastasis. Benign tumor they may be differentiated and they may not undergo invasiveness and they cannot metastasize. They cannot move from one place to another, then they can induce ile hali yake ya neoplasm to the another area of the body. Lakini malignant tumors, they are under unregulated growth and they lack differentiation and they may undergo local tissue invasiveness and often they can metastasize from one part of the body to another part of the body through either circulatory system or lymphatic system. Some benign tumors can be inoperable and perhaps fatal. Japokuwa benign tumors, they are not cancerous, lakini sometimes they can be inoperable and perhaps fatal. Zinaweza hata kuua depending on what kind of tissue zimekuwa. So hata kama ni benign tumor lakini let's say imekuwa kwenye vital organs, let's say brain au moyo au ini au figo, that organ may be damaged, na mtu anaweza hata akapoteza maisha. Lakini mara nyingi benign tumors kama zikaota sehemu kama kwenye mguu au elsewhere au kwenye organ yoyote ambayo it is not vital organs, inaweza tu ikawa cured kirahisi compared to malignant tumors. Let us see the genesis of cancer. Normal cells turn into cancer cell because of one or more mutation in its DNA. This is very important. Normal cells turns into cancer cell because of one or more mutation in its DNA. Kwamba seli kwanza ilikuwa ni normal, then inaweza kuchange kuwa cancer cells kwa sababu ya one or more mutation in its DNA. which can be inherited or acquired. Mtu anaweza akalisi au anaweza acquire through life. And the development of cancer is a complex multi-stage processes involving not only more than one genetic change but also other epigenetic factors like hormone action, carcinogens, and the tumor promoter effect, and so on. Development of cancer ni multi-stage process, sio process ambayo ni ya mara moja. It is very complex multi-stage processes ambayo inahusisha sio tu zaidi ya genetic changes moja ambayo inaweza katokea, lakini pia hata other changes ambayo they are not genetic zinaweza influence formation of cancer cells. Examples of epigenetic factors that may influence or may be involved in genesis of cancer, it's like hormonal action or carcinogens, carcinogenic substances, and the tumor promoter effects, and the other factors like sexual behavior, reproductive behavior, or the prolonged exposure to UV light, and so on. Those are factors that are epigenetic factors that can be involved in genesis of cancer. Very important thing to note, the cell which is normal turns into cancer cell because of one or more mutation in its DNA, which can be inherited or acquired. A normal cell turns into cancer cell because of one or more mutation in its DNA, which can be inherited or acquired. So zipo zaidi ya genetic changes au genetic changes zaidi ya moja ambazo zinaweza kupelekea genesis of cancer, lakini pia zipo other epigenetic factors like hormonal action, carcinogen, and the tumor promoter effect ambazo zinaweza zikapelekea for example of hormonal actions, the epigenetic factors for genesis of cancer is the estrogen and the progesterone hormone for the female sex hormones. They can cause the breast cancer cells to grow. Some types of breast cancer cells have estrogen receptors and the progesterone receptors on the surfaces or inside of the cells. So when these hormones attack to those receptors, they can cause the cancer cells to grow and they divide. So estrogen and the progesterones hizi ni sexy hormones, female sexy hormones ambazo zinaweza zikatrigger breast cancers. Zile cells ambazo zinakuwa found in breast zika undergo ile cancer genesis of the cancer. And some types of breast cancer cells zina estrogen receptors and the progesterone receptors on the surfaces of the cells. So when these hormones, progesterone and estrogen, attack to those receptors, they can cause the cancer cells to grow and to divide na kuwa nyingi zaidi na kusababisha the condition kuwa worse and the worst, and hata mtu anaweza akafa kwa sababu ya cancer. But these epigenetic factors, they are not themselves cancer producing, but they increase the likelihood that the genetic mutation will result in cancer. Zenyewe hazizalishi cancer cells. These epigenetic factors like hormonal actions, sexual behavior or reproductive behavior and so on, they are not themselves cancer producing, but they increase the likelihood that the genetic mutation that will result in cancer. Before niliwaambia very important things to note. A normal cell turn into cancer cell because of one or more mutation in its DNA. So even those epigenetic factors, they are not themselves cancer producing, but they increase the likelihood that genetic mutation will result in cancer. But in considering biology of cancer, we only concentrate on genetic changes and not epigenetic factors. So there are two main categories of genetic change that lead to cancer. Two main categories of genetic change that lead to cancer. These are two main categories of genetic change that lead to cancer. First, activation of proto-oncogenes to oncogenes, and the second, inactivation of tumour suppressor genes. These are two main categories of genetic changes that can lead to cancer. Before tulisema a normal cell turn into cancer cell because of one or more mutation in its DNA, because of one or more genetic changes in its DNA, which can be inherited or acquired. So these are just two main categories of genetic change that may lead to cancer. First, activation of proto-oncogenes to oncogenes, and second, inactivation of tumour suppressor genes. Let us see the activation of proto-oncogenes to oncogenes. Proto-oncogenes are genes that are normally control cell division, apoptosis, and differentiation, but which can be converted to oncogenes by viral or carcinogen action. So these proto-oncogenes are genes which are very important to control the cell division, and also these proto-oncogenes, they are responsible for apoptosis and the differentiation of cells. These proto-oncogenes may be converted to oncogenes by virus and other carcinogenic substances. Carcinogenic substances are the substances which may cause cancer. So carcinogen action ni ile action ya carcinogenic substance kusababisha cancer. So even some virus are carcinogenic, may convert these proto-oncogenes to oncogenes. These proto-oncogenes zina uwezo wa kucontrol cell division na kucarry out this process known as apoptosis and to control cellular differentiation. But once these proto-oncogenes converted into oncogenes, these proto-oncogenes converted into oncogenes, zinapoteza ule uwezo wake wa kucontrol cell division, apoptosis and cellular differentiation. Cell division is well understood and differentiation is well understood, but apoptosis is a programmed cell death process. Apoptosis removes cells during development. It also eliminates the precancerous and the virus infected cells. Although successful cancer cells manage to escape apoptosis, they can continue dividing. Japokuwa zile this apoptosis is a programmed cell death, ina uwezo wa kuremove cells ambazo ziko abnormal au zimekuwa infected by virus, zenyewe zikazi eliminate kwenye system and zile abnormal cells zisiendelee kujidivide. Japokuwa zile successful cancer cells ambazo tayari zishakuwa cancer cells zinaweza zika escape this process of apoptosis na zikaendelea kujigawa zikawa nyingi and so on. But apoptosis maintains the balance of cells in human body and is particularly important in immune system, kwamba these proto-oncogenes are responsible kwa ajili ya ya kucontrol this apoptosis. Apoptosis is responsible kwa ajili ya kucontrol au kumanage ile balance ya cells in the human body. Once a cell is abnormal or infected by virus, through the apoptosis, those abnormal cells or infected cells may be destroyed or removed during development. So once these proto-oncogenes ambazo zina control apoptosis and differentiation pamoja na cell division zikiwa converted by these carcinogenic substances na kuchange kuwa oncogenes, zinapoteza huo uwezo wa kucontrol hizi process tatu za muhimu kwa ajili ya kuhakikisha abnormal cells haziendelei kukua ndani ya mwili. Apoptosis is very important kwa sababu inatumika kwa ajili ya eliminate precancerous and virus infected cells. So once apoptosis fail, the cancer cells may continue divide and grow more and more and more, and the condition to become more bad, to become worse. Activation of proto-oncogenes to oncogenes by carcinogenic substances or virus may lead to development of cancer. Another genetic change that can lead to cancer is inactivation of tumor suppressor genes. Normal cells contain genes that have ability to suppress malignant change. Seli za kawaida za mwili zimecontain genes ambazo zina uwezo wa kusuppress au kuzuia maendeleo ya malignancy. Kwamba zile cancer cells once cells become abnormal, mwili una uwezo wa kucontrol, wa kusuppress ule uwezo wa seli kutaka kuwa abnormal. So the normal cells contain genes that have ability to suppress malignant change, termed tumor suppressor genes. These genes, they are known as the tumor suppressor genes or anti-oncogenes. And there is now good evidence that mutation of these genes are involved in many different cancers. So now there is good evidence kwamba mutation of these tumor suppressor genes or anti-oncogenes, once they are mutated, zinapoteza ule uwezo wa kusuppress malignant change. So thereafter kama zimepoteza ule uwezo wa kusuppress malignant change, tunapatia nafasi ya zile cancer cells ku grow and to divide more and more, na condition kuwa mbaya zaidi. So the loss of function of tumor suppressor genes can be critical event in carcinogenesis. So the loss of function of tumor suppressor genes, once the tumor suppressor genes are inactivated, ule uwezo wa tumor suppressor genes ukishakuwa inactivated, basi carcinogenesis may occur. Carcinogenesis the word the word used to describe the genesis of cancer. Carcinogenesis is the process of production of cancer cells. Let us see the common types of cancers. Baada ya kuwa tumeshaangalia the two common genetic changes that can lead to cancer, one tumeangalia ni activation of proto-oncogenes to oncogenes, pia tukaangalia the inactivation of tumor suppressor genes. Tukasema hizi proto-oncogenes ni muhimu kwa ajili ya kucontrol cell division, apoptosis and cellular differentiation. Once they activated to oncogenes, tunapoteza ule uwezo wa kucontrol cell division, cell differentiation pamoja na apoptosis. So itapatia chance cancer cell to divide na cancer kuwa nyingi zaidi. Na genetic change nyingine ni inactivation of tumor suppressor genes. Kila seli normal seli ina uwezo wa kusuppress ile malignant change development. So once tuna inactivate the tumor suppressor genes, mwili utakuwa hauna uwezo wa kuzuia zile cancer cells zisiendelee kudivide. So let us see the common types of cancers. The name of cancer is derived from the type of tissue in which it developed. The type of cancer is derived from type of tissue in which it developed. So most human cancers are carcinomas. Most type of cancers are carcinoma. This is the first type of cancer that arise from epithelial cells. Melanomas, for example, are cancerous growth of melanocyte skin epithelial cells that produce pigment melanin. So carcinoma is a category of types of cancer that develop from epithelial cells. Specifically, carcinoma is a cancer that begins in a tissue that lines the inner or outer surfaces of the body, and that arises from the cells originating in endodermal, mesodermal, or ectodermal germ layers during embryogenesis. While these melanomas occurs when pigment producing cells that give the color to the skin become cancerous. So as we have already seen, the name of cancer, the name of type of cancer is derived from the type of tissue in which it develops. So those type of cancers occurs in epithelial cells. They are known as carcinomas, and those type of cancers grow melanomas, for example, are the cancerous growth of melanocytes skin epithelial cells that produce pigment melanin. In short, melanomas occurs when pigment producing cells that give color of the skin become cancerous. Sarcoma is a general term for any cancer arising from the muscle cells or connective tissue. For example, osteogenic sarcoma are the most frequent type of the childhood cancer, destroys the normal bony tissue. So sarcoma is the general term for any cancer arising from the muscle cells or connective tissue. For example, osteogenic sarcoma is the most frequent type of childhood cancer that destroys normal bony tissue. And also we have leukemia, is the cancer of blood forming organs characterized by rapid growth of abnormal leukocytes. And also we have lymphoma, is a malignant disease of lymphatic tissue. This is kind of cancer that occur in a lymphatic tissue, for example lymph nodes. This is summary of characteristic of benign and malignant tumors. These are characteristics, and this is benign and this is malignant. Here we see uncontrolled proliferation in benign is we found uncontrolled proliferation of cells in benign tumors, and also we find the uncontrolled proliferation of cells in malignant. And also the benign tumors, the cells in benign tumors, they are not differentiated, and they are not undergo invasiveness, and they are not undergo metastasis. But cells in malignant tumors, they undergo uncontrolled proliferation, they are differentiated, they undergo invasiveness, and they undergo metastasis. So these are basic and important characteristic of benign and malignant tumor. I hope everyone should be able to explain how is uncontrolled proliferation in benign and malignant tumors, and how is the differentiation in malignant tumor, and how is invasiveness in malignant tumor, and how is metastasis in a malignant tumor. Here let us say etiology and associated factors for tumors or cancer. Several factors may trigger a normal cell to lose control and become cancerous. Several factors may trigger a normal cell to lose control and become cancerous. One cause is environmental agent, like substances in the air we breathe, water we drink, and food we eat. So vile vitu tunavyokula, hewa tunayovuta pamoja na maji tunayokunywa as environmental agent may be one of the several factors ambazo zinaweza zikatrigger normal cell to lose its ability to control the cell division and become cancerous. And also a chemical agent or radiation that produce cancers is called carcinogens. So na zile chemical agent or radiation ambazo zinaweza zikaproduce cancer cells, they are known as carcinogens. So carcinogens is any cancer causing substance. Any substance that can cause cancer is known as carcinogens. Let us see the three classes of carcinogenic agents. First, we have chemical carcinogens, and we have radiant energy carcinogens, and also we have microbial agent carcinogens. These are three important classes of carcinogenic agents. Chemical carcinogens. Chemical carcinogens directly damage DNA, leading to mutation and eventually cancer. Direct acting agents do not require metabolic conversion to become carcinogenic, while indirect acting agents are not active until converted to an ultimate carcinogen by endogenous metabolic pathways. Chemical carcinogens moja kwa moja zina damage DNA. Before tulisema kwamba ili mradi cancer cell itokee idevelop, lazima kuwepo kuna one or more DNA changes. So these chemical carcinogens moja kwa moja zinaenda kudamage DNA, leading to mutation and eventually cancer develop. Sasa hizi chemical carcinogens zipo za aina mbili. Zipo ambazo ni direct acting agents na zipo ambazo ni indirect acting agent. These direct acting agents, they do not require metabolic conversion. Hazihitaji kuwa converted ili mradi ziwe active ili kwamba ziweze kusababisha cancer. Zenyewe tu they are direct acting agents, zina uwezo wa kusababisha cancer hata bila kuhitaji any activation kama metabolic conversion. Lakini hizi indirect acting agents are not active. They are not active until converted to an ultimate carcinogen by endogenous metabolic pathways. Ndani ya mwili zipo enzymes ambazo zina uwezo wa kuconvert chemical substances from its inactive form to become active, or from its active form to become inactive. So these enzymes known as cytochrome P450, cytochrome P450 may be used to activate these indirect acting agents to become carcinogenic and they may cause cancer. So chemical carcinogens may be either direct acting agents or indirect acting agent. This direct acting agent, they do not require any metabolic conversion to become carcinogenic. Lakini is indirect acting agent, they require metabolic conversion to become carcinogenic. So this polymorphism of endogenous enzymes like cytochrome P450 may influence carcinogenesis. Carcinogenesis. These are enzymes are found in almost every tissue in our body, but they are highly concentrated in the liver and other organs like heart and so on. So and other organs like brain and the liver. These enzymes cytochrome P 450, they are found in almost in every tissue in our body, but they are highly found in the liver. Zinaweza convert hizi indirect acting agents na kubecome carcinogenic na kusababisha cancer. Then following exposure of a cell to mutagen or an initiator, tumor genesis can be enhanced by exposure to promoters which stimulate proliferation of the mutated cells. Mutagen is any substance that can cause mutation. So following exposure of a cell to mutagen, anything that can cause mutation or an initiator, tumor genesis can be enhanced by exposure to promoters which stimulate proliferation of mutated cells. So zile mutated cells ambazo ziko mutated cells ni cell ambazo zishakuwa mutated, ambazo mutation zimeshakuwa taken place inside. So once a cell umeexpose kwenye mutagen, mutation imeshatokea, then tumor genesis can be enhanced by exposure to promoters which stimulate proliferation of the mutated cells and the condition to become so worse. Examples of human carcinogens include direct acting like alkylating agent used for chemotherapy. These are drugs used for the treatment of cancer. They can also induce ile carcinogenesis na cells zikaendelea kujigawa without control and zikawa abnormal and so on. And other indirect acting example aflatoxin. Radiation carcinogenesis. Ionizing radiation causes chromosome breakage leading to genetic damage and carcinogenesis. UV X-rays give rise to squamous cell carcinomas and melanomas of the skin. Ultraviolet light from the skin from the sun may lead to carcinomas and the melanomas of the skin. And the microbial agents, oncogenic viruses, humanity lymphotropic virus type 1 causes T-cell leukemia, and the human papilloma virus has been associated with benign warts as well as cervical cancer. These are viruses that may cause cancer. And the bacteria, Helicobacter pylori, is strongly associated with development of gastric cancer. The tumor marker is a substance introduced into circulation by tumor cells that indicate the presence of tumor as well as a specific type. So tumor markers may be used to screen, diagnose, make prognosis, evaluate a response to treatment, and monitor for recurrence of cancer. These tumor markers, these are substances, especially proteins, produced by body or even in the cancer cells, and once introduced into the bloodstream may be detected either in the bloodstream or in the urine, na give indication kwamba somewhere kuna cancer cells zimekuwa produced. So tumor markers may be used to screen or to diagnose or to make prognosis, to evaluate a response to treatment, and monitor for recurrence of the cancer. So here are key points, very important. Everyone should make sure anapitia hizi key points kujiridhisha na kuhakikisha kwamba amecover sehemu zote muhimu. So cancer is a group of disease characterized by uncontrolled or abnormal cell proliferation. When cells in a part of the body divide without control, the excess tissue that develop is called a tumor or neoplasm. Hii tulishaiona above kabla. and the 3 classes of carcinogenic chemicals: radiant, innate and microbial agent. Kwenye chemicals tuliziangalia kuna direct and indirect agent, and kwenye radiant innate tumeangalia UV light, and microbial agent tukaangalia HPV pamoja na H pylori bacteria ambao wanaweza wakasababisha cancer. This is evaluation. Everyone should take a time to evaluate him or herself, kuangalia kama anaweza kujibu haya maswali yote. What is cancer? What are the common type of cancer? What are the characteristics of benign and malignant tumors? And what are the factors associated with the tumors or cancers? This is the last slide of our presentation. Thank you for listening.